everyone! For viral infections to take place, the first stage of the life cycle is the virus attaching and entering into the host cell. However, viruses don't just enter any cell. It is only tropic for certain cells. Only certain cells will provide the specific receptor molecules for the virus to attach, so only certain cells are susceptible. Additionally, only certain cells will allow the virus to transfer its genome into the cell and contain the growth factors to support viral growth. In other words, only certain cells are permissive. For the viral infection to take place, you must demonstrate tropism, i.e. permissivity and susceptibility. And one essential part to understand about viruses is that they enter by receptor-mediated endocytosis. This process is not passive pinocytosis because the virus particle is much bigger, and it is not phagocytosis because it doesn't usually involve immune cells. Once the virions bind to the receptors, this drives a conformational change. And this conformational change may expose fusion regions within the virus, allowing it to bind to endosomal membrane. Then the endosome becomes more acidic, and this leads to uncoating and disassembly of the capsid. Fusion proteins can be divided into two groups, class 1 and class 2. Class 1 fusion proteins are usually expressed as a trimer, and it is expressed as a precursor, which is cleaved when it is moved to the cell surface. And cleavage causes activation and increased functionality. It has its C-terminal anchored to the viral membrane and the N-terminal containing 20 hydrophobic amino acids to bind and insert into the cellular membrane. Consequently, it drives like a pore formation, and the fusion protein is exposed and inserted. Examples of viruses which use class 1 fusion proteins include SARS-CoV-2, influenza, HIV-1, and Ebola. Class 2 fusion proteins are predominantly non-helical, sheet-like, and they don't need any cleavage for activation. They might just need some sort of activation signal through pH, in the case of dengue virus and the protonation of histidine residues. Lastly, from this lecture, it is important to remember that you can differentiate viruses from those which only use receptor-mediated endocytosis and those based on direct fusion by using a weak base. A weak base such as chloroquine and methylamine would block receptor-mediated endocytosis but not fusion. And it is also important to remember that viral components move through the cell through the use of microtubules such as actin. Vaccinia, which is a pox virus, can use the actin on filopodia to move to the cell body and induce membrane blebbing. Thank you so much for listening. See you next time.